All right, so you've heard from each of our panelists. The rest of the night is up to you with audience questions. I have given them two instructions. One is to talk directly into the microphone so we can all hear them. And the second one, for those of you who have been here for previous science cafes, they are supposed to repeat the question into the microphone before they answer. Now, that's always a problem because you get excited about answering the question, but last month was especially fun because our topic was memory and they could never remember to repeat the question. So our goal is to do better than the memory folks, which will be very entertaining. All right, so who's got a question? And it can be for any of these people. Yes, ma'am.
Dr. Ussery. So is somebody doing something with all of this terabyte that you're putting into whatever you're putting it into? It's not just sitting there, I hope.
No,
Repeat the question.
yeah, okay, so it's someone doing something with all these terabytes of information. And in fact, I have a dozen people in our group. We have a new cluster, new computer that we just got, and we're really kind of wanting to be able to get in and use it. And in fact, I came here from Oak Ridge National Labs, and they had big computers there. When I moved there, it was the biggest computer in the world. And we're working with them. The reason I went to Oak Ridge wasn't because they had big computers, but because they had smart people that knew how to use big computers. And you know, lots of people can generate lots of data. Lots of people can have a need to use a big computer, but do you really, are they going to do anything useful, right? And I'm all for, I keep telling my students, it really helps to have a clue. It really does. And so to give you an example of something useful, so I told you about this, a lot of these results we get. We have 100,000 bacterial genomes that have been sequenced. Now to compare the proteins on this, if you extract all the proteins, there's 300 million proteins. To compare them against each other, you know, you can go to Amazon Cloud and you can spend some time on it. It would take more than 1 million years, and it would cost $120 trillion. And I have, so Visnu, he's hiding in the back, he couldn't get in, but we have a programmer that we've hired. He basically compared those 300 million proteins, and it took him 2 days, using a small set, basically 10 CPUs, because he knew what he was doing.
Yeah.
It really helps to have a clue. And so, you know, it's easy to use a lot of computer time and waste a lot of energy, but it helps to actually know what's going on.
Another question? Yes.
You said you had been to Norway.
Just came back from Norway, from Trondheim. I'm a 20% professor there.
We have type 1 diabetics in our family, all our children, husband. So we've been hearing that you are doing some things in Norway with microbiome relating to children becoming diabetic, juvenile diabetes. Were you working with that at all?
I was not working with that project, no.
We repeat the question.
Oh, the question was, are we doing anything with Norway on diabetics and the microbiome? And the answer is no. I was in Denmark part of this MetaHit project. We did look at diabetics there. That's kind of the same idea. And probably these guys,
Yeah, I mean, it's many of the childhood diseases, especially the diseases that involve the immune system, you know, seem to be dependent on the microbiome. And what's really kind of fascinating is the studies that have shown how you can manipulate the microbiome to change this. You know, there's several studies, of course, using probiotics, of course, probiotics being in very early stages now. But it really seems like the critical time when you can change the microbiome is almost the first month of life. We know that within the first years of life is when the microbiome is flexible and reaches adult levels after a few years of life. But it seems like the time that's critical for actually manipulating the microbiome for many of these diseases, allergic diseases, asthma, etc., seems to be almost in the first days of life, the first 28 days of life. And the other thing that's really fascinating is, you know, that we're finding that the microbiome actually develops before birth. That, you know, that the amniotic fluid, which we thought was sterile, actually has its own microbiome. And it's important because the type of birth, you know, whether you have a C-section or not, depends on how the microbiome is actually transferred from your mother to you, and how your microbiome develops in that critical time of life. What happens to you after birth if you're sick and you go into the ICU or get antibiotics? That can completely disturb your microbiome. So understanding these things allows us to actually correct things early on in life so that we can improve health, especially what seems to be a lot of immune diseases, and actually make a more healthy population.
And just to add to that, and Richard, you may know, someone told me earlier today that there was someone in the Department of Pediatrics at Children's named Sean somebody, Sean Adams, who looks at the microbiome and diabetes, but I don't know if it's type 1 or type 2. I don't know that, but that would...
Type 2.
Type 2? Okay, all right, sorry.
What was his name again?
Sean Adams.
Thank you.
Yep, thanks. Yes?
Dr. Fry, are you familiar with the GAPS diet and gut and psychology syndrome diet, and how do you feel that has any validity in a lot of the things you looked at?
Repeat the question.
Okay, am I familiar with the GAPS diet and I'm sorry, what was the other diet?
Well, that was just, what it stands for is the gut and psychology syndrome.
Oh, okay, the gut and cytology syndrome diet, obviously not. But I'm familiar with a lot of different diets. Mostly what they're composed of, how they're theoretically supposed to manipulate the gut. I think there's many different theories. We are very big proponents of diets, especially elimination diets. We find these in our children with autism that there are certain things that they're sensitive to that aren't actually within the framework of allergies, which I think is a big misconception. Everybody thinks that, well, if you can't eat something, you must be allergic to it. And that really puts a context of a certain physiological mechanism. And we're finding out that probably people are sensitive to all types of environmental agents, and they're not these pure allergens that we think of. I think it's opening our doors to understanding how to make us healthy. Now, for children with like autism that seem to respond to some of these diets, there's many reasons why. Now, it can be sensitivities to food. It also can be the fuels that you're giving. The microbiome, the bacteria, and such. And there's many other reasons. We're very interested in diet, especially since we work in metabolic disease. So both nutrients and the fuels that you give to the body will, we know, manipulate and can change the way metabolism works. The problem with a lot of the diets, and not to be critical of them, is that there isn't a lot of great, good clinical trials on these, and that's not to say that people haven't tried. Diet is something that's very, very hard to study, and because usually when we do a study, we do a double-blind placebo-controlled study where we have a placebo, which looks like. Now, diet is very different, and then the other thing that you have, and that I think that we're learning, and I think this is some of the work like you're doing with genomics and personalized medicine, is that everybody isn't the same. So we're also used to looking at things and studying them as if everybody is the average. And we're not. We're all individuals, and we respond many different ways. So I think diet is very important for many different reasons. And what we find is that, many children we find may respond to different diets, and it may be for different reasons. One of the challenges is to find out how to study that, how to measure it, and then find out who is the person that will respond to each of those different diets. So I hope that answers your question.
Thank you. I think that was developed for kids with autism, and that's the reason I had read
Yeah.
something about that.
Yeah, I mean, and there's many different diets. There's the GAPS diet, of course, the gluten-free, casein-free, diet. And there's many other different diets, and many times what happens is that it's very difficult to know why a child may actually respond. So many times we suggest that in the context of a nutritionist, and this is very difficult also because a lot of times we don't have people, since it's not very well studied, there's only so many nutritionists around the country that really know how to guide people correctly on these diets. So it becomes something that's very difficult to do, and I applaud parents that try and to do this. Of course, it's important to have a nutritionist on board to make sure that your child gets the right nutrition. And if they don't, there's ways of supplementing that. And that's when they come into our clinic, that's one of the things we look at. If they're on any special diet, we want to make sure that they are getting the right nutrition, in addition to the diet. But I think we very much recognize that that diet is very important for many different reasons.
Yes, sir.
I'd like to learn a little bit about the mechanisms by which bacteria influence the immune system. Are the cells engulfed by the human cells, or is it metabolites from the bacteria? How does it work?
It's
Y'all can all take a stab at that. We can just go down the road.
so how does the bacteria influence the immune system? Bottom line is we're not sure. Okay, I think it's very early on, but in many ways, a very large amount of your immune system is actually in your gut. You actually have lymphoid tissue in your gut, and you actually, and as you grow very early on, your immune system actually grows in harmony with those bacteria. And in that sense, it develops tolerance to certain bacteria and finds out which are the bad players. In that sense, it can actually program some of the cells. That's one mechanism. Another mechanism we're finding out is that there's certain mediators that the bacteria can release, and that actually manipulates and can make the immune system increase or decrease. And there's many different mediators, and one of the things that we're finding is that different balances in these different mediators will actually change the way the immune system reacts. So that's another way. Another thing that we're finding is that a disturbed microbiome in many different ways regulates intestinal permeability. And intestinal permeability is very important because usually, you have a very tight gut barrier, and if you don't have that, then certain types of molecules, certain types of proteins that would not get in and be exposed, your immune system wouldn't be exposed to, is now exposed to it, and it starts to get sensitive to things that it wouldn't actually see, it wouldn't actually have to get sensitive to. So there's many different levels, and these things can happen at many different times. One of the things that's also very fascinating is some of the studies that look on what a healthy microbiome is. We like to think of what are good players and bad players, and one of the things is we find that there's some way of saying, okay, these may be good bacteria, these may be bad bacteria. One thing we're finding is that a diverse microbiome, one where you have many different players, and so you have like a very rich forest, of all different plants and trees and fauna and flora in there, is actually something that stimulates the body in the right way. And that when you have only a few players in there, that is representative of a very sick microbiome. Some of the studies that have looked at microbiota transfer, or fecal transplants, are really interesting because they've looked at if you transplant somebody else's, and I assume everybody knows what a fecal transplant is, or and so what
Why don't you explain it a little bit?
So just
Not
there are two
fully
ways.
graphic terms, but sort of a broad view.
So that's why we've created a new word called microbiota transfer therapy instead of to make it a lot softer, But essentially, you take the substance from some person's gut and you put it into another person's gut. Of course, you can imagine how you get that substance. But anyway, so what we've been able to do is we've been able to refine this technique so that we can more easily and transfer that microbiome from one person's gut to another. And it's a very complicated process. And in many ways, you know, we started out and people still, you know, use very complicated techniques where they might have to put a tube down to actually get through the stomach and put the substance in, or use an enema or something. But now actually technology is advancing, so it actually can be in a form where you may be able to take it like a regular medication. And the technique, the first thing you do is you usually give a very strong antibiotic, usually vancomycin, which is something kind of, you can think of it as just nuking your gut. So you get rid of all your bacteria, and then you put somebody else's bacteria in there. And it ends up that many times, and it seems like, especially if your microbiota, your microbiome is very disturbed, that putting somebody's new microbiome in there actually can stabilize and actually can fix the problem. And this has been pioneered in something called C. difficile infections, which are extremely bad infections, very, very hard to treat. And so it's almost standard therapy at some hospitals now. And now we're starting to look at other things like diabetes and other types of disorders where you can do this. But one of the really interesting things is when they did this type of therapy, and then they said, well, what happened? What is the, we've transferred, you know, person's A microbiota into person B. Now this person's B, let's take a look at their microbiome. Does it look exactly like person A? Ends up, no. No, it doesn't. It sets up its own life. But what it does is it creates the seed of diversity, to increase the diversity in the gut, so that if it grows correctly within the intestine, it actually sets up a stable ecosystem, and those are kind of the seeds of the ecosystem. So it's very complex, and it's very interesting in that sense. I hope I answered the question.
So Ruud, do you want to talk about the immune surveillance and tumor microenvironment?
Sure. So I do not work with patients. I have the fortunate situation that I can manipulate my mice in a certain way. So coming back to how does this actually work, what is the interaction? Fortunately enough, the early studies showed that if you grow a mouse after weaning, or just after weaning in a germ-free facility, the immune system is not completely developed. And so therefore, those were the essential papers which really got me interested to study it further in terms of cancer as well, showing that indeed the symbiotic nature of the microbiome with the immune system is so essential. And then we learn more and more on how many levels. And some of them Dr. Fry already alluded to. In my lab, we are indeed inducing dysbiosis or perturbance of the microbiome by low-dose antibiotics, one of which is also vancomycin. And we are studying cytokines, so these are messenger molecules from leukocytes which can talk to each other or to other cells. So that's what our studies are focusing on, how this crosstalk or systemic signaling between the microbiome and our interest then is beyond the cancer, is actually the vascular of the tumor itself, how that interacts. So we see that if we perturb the microbiome, we have a level of dysbiosis, certain cytokines are downregulated, and therefore the immune system does not respond as it normally should. And then our particular, we focus then on a particular mechanism on the endothelial cells or the inner lining of a tumor blood vessel, and how they respond and which molecules are involved.
So I have a granddaughter that has tuberous sclerosis. Are you familiar with that?
A little bit.
So we give her a real expensive probiotic, it's like $100 a bottle. Do you think that we give it to her because of autism, but do you think that would possibly keep the tubers from growing?
I would not be able to answer that question. That would go beyond. Also, the question is, her daughter or granddaughter has tuberculosis. She's getting a probiotic, and she's wondering how this would potentially influence or prevent cancer. I'm not.
It's not cancer, it's the growth of the tumors.
Okay, the growth of itself. Yes. Okay. At this stage, it's way too early for me to say something significant about it, and I'm not a physician, so I cannot guide you in any of that.
Thank you.
Okay, so Dr. Fry, as you know, our little ones with Down syndrome, and obviously we got some autism stuff going on. But a lot of our kids are behind the 8 ball from heart surgery and the high doses of antibiotics. And we do the elimination diet, we do everything. Is there an effective way to test our kids' gut bacteria? I mean, we can do stool samples, but how, are they accurate,
Might you have some more insight into that?
So-
Repeat the question if you would, please.
So the question was whether there's effective ways to look at gut bacteria. And, we have to say this because we've been talking about fecal samples. People give you shit for free all the time. You don't even have to ask for it. So we have lots and lots of free samples from people. And in fact, even one of her postdocs, she said an anonymous, okay, we know where it came from. Anyway, so we have lots of fecal samples. And one of the things that we're looking at, most people, and probably most of the stuff you're doing, 16S, right? And there's something, the method that's been around for many years called 16S ribosomal RNA. It's a very well conserved gene that's in all bacteria. It's in all of life. Yeah, 16S ribosomal RNA, and it's called just 16S sometimes for sequencing. And the analogy I use is, okay, you have a picture of Yellowstone National Park. And you have this picture, and it's kind of, you can see some blue in the sky, but it's kind of really more abstract, painting. And you can see some green grass, and you see a little brown spot, and that might be a bear, but you're not sure. And you see something that might be some mountains. But it's a really low resolution. 16S gives a very kind of low resolution view of the families that are involved, of families of bacteria. But it doesn't get down to the genus species, to like the first and last name. So imagine you have a population, in Arkansas, you say someone comes from this town, that's kind of okay, but a lot of people come from the town. But if you know the name of the family, you know the first name and last name, you know who it is. So these things, let's see, now Intuat.
It's like $2.50 a piece.
So Intuat tells me that I should actually ask for some money from this company because I hug it, but it doesn't have to be this, but there are new technologies where you can sequence all of the genomes, and part of the reason people haven't done it is historically it's certainly been expensive and also difficult computationally. But one of the things in Denmark we looked at, part of an EU project, 200 people from Denmark, 200 from Spain. Like I said, we get free fecal samples, and we have time points, and so then we have thousands of samples, and then from that we can reconstruct the population. And exactly like Dr. Fry said, that diversity is really important. So the healthy people have lots of different types of bacteria. And then the people that are very sick, a lot of times they have like Crohn's disease and other stuff, the diversity is very low. So there are more accurate methods, and we're working on trying to improve them, and I'm part of something called Genomic Standards Consortium. We're trying to get some standards because there's a disagreement on names for things as well, even. So for example, we talk about C. diff infections, Clostridium difficile. Well, the people, I joke it's the Russian mafia, they renamed it Peptoclostridium. I don't know if you remember this or not. And then it turns out it's a different bacteria. It shouldn't really be called Clostridium, it's Clostridioides now, difficile. But from a medical doctor's point of view, they don't care, they just C. diff, right? C. difficile, you need to know the name. And the names are important. And I'm digressing, but the point is that if you have the genome sequence, that's the best identifier. Then you know specifically who's there, and you can get a really, really good picture. Now, the problem is, if we're to do that for these people, right now, because we don't have a baseline, you get this really, really super sharp picture, but you have no idea what it's meaning. And so we're going to have to do this for lots and lots of people, and then figure out, okay, these bacteria are normal, these are not, and there's, for a typical person has 400 or 500 different species of bacteria living in their gut. We really only know the first and last name for less than 100, probably 50. of those. The others, some of them, we have no idea even what phyla they belong to, which is a pretty high level of classification. You know, we don't know whether they are, you know, a plant or an animal, that level.
And one of the other things, I don't know if you would agree with this, but, you know, going to your question about, you know, the level of detail and you know, how to measure that, I think also is the question of what is the right metric, right? And this is stuff I think Intawat is working on, you know, we're actually looking, you may not need to know the first and last names of everybody. You may need to know how they're organized, how diverse they are, you know, how many different cultures there are within there, you know, and that's something that I think we're still learning, so we understand what's a stable microbiome that's resilient, you know, because you give one person antibiotics and they're fine, you give another person antibiotics and they're not. So which are the resilient microbiomes, which aren't, and then how do we preserve that? I mean, can we look at their microbiome to begin with and then understand which is the right antibiotic to give them, not to disrupt them, or which is the right probiotic to give them afterwards, or which is the right diet, you know, to give them to make sure that their microbiome stays stable.
Yeah, so it's not just what is their name, but also what do they do, is another way of saying it in terms of, yeah.
Yes.
So most of the stuff we've been hearing is how subtle to modest changes in the microbiome predisposes to various outcomes, whether it's disease or... You know, people who've had cholera, how can you imagine, would be one of the most drastic changes in the microbiome. Has anybody looked at those people to see whether they're susceptible to any disease? So that's one of the most drastic changes you find in the gut microbiome after a bout of cholera.
So the question is, what happens if someone has a drastic change? For example, cholera could change the microbiome. Certainly in Haiti, remember a couple of years ago, there's an earthquake in Haiti, and then there's an outbreak of cholera. Now, before that, it had been 100 years, more than 100 years, no cholera in Haiti. So what happened? Well, it turns out that one of the things, in order for cholera to grow, the water needs to be a little bit basic. And it turns out that the streams in Haiti were acidic. But in the earthquake, big, think about it, there's these big limestone caves. And limestone is essentially calcium carbonated, right? And what happens is now the water switched from being acidic to basic. So now this bacteria that normally wouldn't grow in the waters could. There's this big controversy. You heard about the United Nations peacekeepers. And I had a Danish student of mine, a guy I worked with at the Center for Genomic Epidemiology in Denmark, works with World Health Organization. And so he got some of the strains of cholera from the Nepalese peacekeepers. Well, some strains of Vibrio cholera from Nepal. And the question is, did it come from the UN peacekeepers? And so I'm sorry, this is a little bit technical, I know, but anyway, the bottom line is that through using genomic sequences, we could actually, so someone had said it's just an environmental strain, and we said it's probably not, that there is this outbreak strain. And the environmental strains didn't have the toxins, but these particular epidemic did, so we can show that. But we looked at, us and several other people, looked at fecal samples from people that had cholera, and you see lots of different things in there, salmonella and other things. And then we can actually, in some cases, they looked after exactly what you're asking. And although we don't know for sure, everyone's individual, it looks like after this, the microbiome kind of turns back to its normal. So a lot of people, you perturb their microbiome, something happens, you take antibiotics, and then you stop. And it kind of comes back to the equilibrium. Because your microbiome is actually kind of determined by a lot of things, by the food that you eat, and also by the counters. They actually have done the microbiome in the home. And so you can look at people's houses, and the microbiome at the lab, and the microbiome of hospitals. You've seen where they look at different things. And there's all these different bacteria that are there, and it's kind of reflective of the environment. So I think you perturb it, keep your fingers crossed, in principle, if you're lucky, you might be able to go back. Don't know.
I have two questions. One is really simple-minded, maybe the second is a little less so. When you speak of the microbiome in the gut, do you mean the stomach, the small intestine, and the large intestine, all three. Yes. Okay, the second question is, if you have an unhealthy microbiome, would that possibly both suppress the immune system and promote the growth of cancer, and or alternatively promote the growth of immune cells and cause autoimmune diseases? Can it do both?
So while y'all are figuring out how to rephrase that, the first question was, as we're talking about the gut microbiome up here, does that include the stomach, the large intestine, the small intestine? The answer was yes. So we're looking at the whole picture of the digestive system. So you all can.
So yeah, and to get to you about the microbiome, when we talk about the microbiome, a lot of times we concentrate on the gut, but the microbiome actually means all the bacteria all over your body. You have a skin microbiome, you have a lung microbiome, yes, we call it the enteric microbiome, the gut microbiome, and that refers to the whole digestive tract. And actually, you have a good point of, which part are you talking about? Because each one of them tends to be, different parts of the digestive tract have distinct microbiomes which operate in different ways. So it's a good point that, we talk about the general microbiome, but we could get more specific. And then I think the second question was, would disruption of the microbiome cause things like cancer and autoimmune disease, and how does that happen?
Was it by both either suppressing the immune system or promoting the immune system too much?
It's so from what I can tell you, it's a very complicated question, because what we see, like a lot of things, is that the immune system can be what seems very paradoxical. A dysfunctional immune system that doesn't work and doesn't attack the right bacteria to protect you can also be the same immune system that attacks your body unnecessarily. So it depends on the different type of way that the immune system is disrupted. The immune system is very complex, has many different cells that control that controls itself, and has many different cytokines, as I think we're alluding to. And these different cytokines, these different messengers, can actually change the balance of the immune system one way or the other, and make it dysfunctional. And the cytokines that we know about and how they work is very complicated, and I still think there's a lot of research still being done to kind of understand what those different messengers are and how they can control the immune system. And the gut actually can change the way that the immune system sends its messages and which messages it sends. And that's what we're trying to understand, I think, is what are those changes in those messages and how that affects the body. And it may be different in different people. You would probably, it sounds like a lot of your research.
Yeah, it would be different. I would agree with all of what Dr. Fry said. And for us, because we can manipulate the system at any time we want, because we're working with mouse models, it's also about the timing of the disruption as well, which makes the complexity even more difficult as well.
Yes, ma'am.
Okay, we've talked about how sometimes antibiotics can destroy the immune system as far as the gut and whatnot. But can you tell me about the pros and cons of immune modulating therapies like Ivig and their effect on the gut? And that goes for any of you.
So that's another big topic, and it depends on the. So let's take something.
So you repeat the question.
Okay. So the question is, what does immune modulating therapies? What is their effect on the gut, and how does that affect the immune system? I think you can think of the gut and the immune system as being in balance. And so when you change the immune system, you may be able to change the way the microbiome affects the immune system and modulates the immune system. but you have certain types of therapies such as IVIG. IVIG can be given in many different ways. IVIG is given intravenously, but you can also give immunoglobulin subcutaneously or orally. And we find out that in some cases, just oral immunoglobulins actually help the immune system and help stabilize it. The other therapies that modulate the immune system, may or may not have certain effects on the microbiome. One of the things is certain types of immune disease. And we're finding that there's certain different signatures to the microbiome that are associated with different perturbations in the immune system. But we're not sure which is the chicken and the egg too, because they're in balance. So if you have a disturbance to the immune system, it may disturb the way that the microbiome develops or what type of microbiome you have, whereas the microbiome that you have can also modulate the immune system. So I think it's very complicated when we get into those type of things, and that's why many times immune modulating therapies can be, I'd say, in many ways less than desirable, because they're kind of very blunt instruments in the way that they change the immune system. I mean, there's some therapies, especially in cancer, that are being more targeted actually to certain parts of the immune system. But some of those general, therapies tend to kind of change things in an overall way and hope that the underlying system actually reorganizes itself and balances out.
Yeah, I will do a follow-up on that. Actually, in the cancer field, we are now actually finding that some of our traditional, conventional, old-school chemotherapeutics are actually immunomodulators. And one of the seminal papers were showing that cyclophosphamide, which was already described to have other mechanisms, actually needed a relatively healthy microbiome in order to exert its activity. So it had an immunal component to its mechanism of action, which patients have been treated with all along. Additionally, there was a follow-up paper by that same group, and her last name is Zitvogel from France. She showed that immunomodulating the newer immunotherapies called checkpoint inhibitors, actually CTL4, it's an anti-CTL4. That therapy will not work if you have a certain modulation of your microbiome. So we are just at the tip of the iceberg, and a lot of these things we do not know what's the chicken and what's the egg, and are these chemotherapies actually working because of the immuno aspect they have, which they have not been described, but they've been FDA approved, so they're being used. So we're just finding that out, and that might be eventually where I think our field is going. Why does a certain cancer patient respond to a certain therapy, and why not? And we're hoping to find more answers and looking at the microbiome that might help us out trying to predict, or how people refer to as precision therapy or targeted therapy as well, to get a better handle on that.
So for a relatively healthy person, and I don't know the answer to the question, can you each talk about the industry or supplementation of probiotics, what that is all about, and is there merit?
So what do you think about probiotics? Do you recommend them? Do you use them? What is that?
Probiotics, we definitely recommend probiotics. We try and stay with brands that we believe are high quality. There's many different probiotics. The problem but there's a lot of issues with probiotics, why they may or may not work, and I don't think we really know completely. One of the most obvious things is that when you take a probiotic, if you're taking some organisms, they have to get through the stomach, which is a pretty toxic environment. Two, different probiotics have different mixtures. Some of them may be more viable than others. So it's very variable. What is the correct mixture of the probiotic to take is a big question. When you take a probiotic, you take a prebiotic with it to give the organisms the right fuel, to work. These are very largely unanswered questions. Probiotics in general seem to be for the most part safe. They seem to, sometimes there's evidence that in some cases they can help with health, but I'd say the evidence is not that strong. So it's very difficult to say definitely, take this probiotic, don't take that probiotic. One of the things that is the advantage of probiotics is that for the most part they're safe. So you can't do much harm. You can just kind of waste a lot of money on probiotics. But they definitely have some use. I think that it's that as we start to learn more and more, as the science gets ahead, we can start to learn what are the correct treatments. How do you make the right probiotic? One of the other things that we worry about with probiotics, as I had mentioned, is that one of the things we're finding is diversity is the key. Well, what happens to somebody that takes the same probiotic for years? Is that helping their gut, or is that decreasing the diversity of their gut? So I think that there's a lot of questions. I don't think that there's strong evidence to say that for this condition or that condition, you need to take probiotics. But on the other side, there's not much harm you can do by taking them.
In the back, yes, sir.
Did our biome evolve along with us in the sense that are there certain bacteria that only can exist in the human gut, and is our biome similar to other mammals? Do we know that? Answer to that.
Actually, so I had some friends of mine.
So you repeat the question.
Oh, so the question is the microbiome evolving with us, and how does it look in other animals? And there is evidence there are some differences, but the differences, from my perspective, aren't that different between one human and another, or as big as between humans and cats. So in other words, the cat microbiome is going to be different than us, but maybe not that different. I think that it would be, certainly the cow microbiome would be different, because if you think about it, the cows, they have 2 stomachs and all this other thing. That's going to be a lot different, right? But maybe a bear microbiome, a monkey microbiome. I don't think it would be that different. I mean, but it is kind of a complicated ecosystem, and at the level that most people, when they talk about microbiome, they look at fecal samples and they're looking at 16S. I don't think there's that big of a difference. That's my own opinion. I don't know what these other guys think.
Yeah, I think one of the things that's interesting, though, is your question about evolution of the microbiome, whether it's evolved with us. And one of the things I think that's really interesting is the recent studies that shows the de-evolution of the microbiome, and that the microbiome, just within a few generations, can be de-evolved to become less robust, and it's very hard to bring it back to where it was.